Regeneration is a great biological advantage to have, so why can't we regenerate whole limbs or organs? Many animals are believed to have had this trait previously and since lost it through evolutionary time. Some organisms have kept their regenerative ability, however, because it is necessary to their fitness. Adult echinoderms are not only able to regenerate new limbs, but can also form new adults from the detached limb. There have been many cases where detached body parts have survived for long periods of time and can still undergo partial or even total regeneration. Regeneration is seen in other species as well. Axolotls have the ability to regenerate limbs, tails, retina, spinal cords, and even sections of their brain. The process of regeneration begins in axolotls immediately once the tail is removed. A blood clot quickly forms and distal muscles relax, losing glycogen content over time. This is called the primary immune response and starts the process of wound healing. This response is faster than seen in mammals, which typically takes up to 2 to 4 days. The immune response attracts white blood cells to the wound site, controlling both pro- and anti-inflammatory signals, as well as promoting dedifferentiation of cells. Keratinocytes from proximal scales and epithelial cells converge at the wound site and a shiny mound of skin forms. This clump of cells is called the blastema. Upon formation of the blastema, a rapid lengthening period begins. A basic ependymal tube is formed to serve as a rudimentary spinal cord, while a cartilaginous tube grows around it to act as vertebrae. Blood vessels develop, causing vascularization to improve and allowing growing tail cells to process glycogen again. Nerve cells branch off from the central ependymal tube and spread out to the distal ends of the tail. Muscle cells start growing out of the line of amputation next to the existing muscle. Elongation and cell differentiation continue for approximately 5 more weeks. At this point, the tail enters its final stage of regrowth, known as maturation. During this phase, scales cover the tail, blood vessels properly vascularize the area, and nerve cells become fully myelinated. The fully regenerated tail will physically resemble the original, save some color variation and a weaker structure. Its simplified structure allows the tail to reform more efficiently. The potential for regeneration is also present in humans, but at a very limited capacity and typically at a young age. Children have the ability to regenerate fingertips lost at the most distal joint. This is observed because children have high local concentrations of insulin-like growth factors, IGFs, and bone morphogenic proteins, BMPs. IGFs are necessary for building an extracellular matrix, which helps organize cells for healing. BMPs induce bone and cartilage formation. These factors are necessary for wound healing and regeneration of new cells. While they are found in adults, they are not present at these concentrations. Children may possess additional necessary growth factors or signaling pathways that are lost in adults that allow them to regenerate their fingertips. The future of regenerative biology is vast and unknown, but hopefully by understanding how regeneration of full organs works in other species, can these concepts be translated into human medicine, and someday maybe we'll be able to grow more than just the tips of our fingers.